Dear colleagues, welcome to EHJ Today. I'm Filippo Crea, Associate Editor of the European Heart Journal. I am in Rome for the ESC meeting, and I'm sitting with Professor Jansen, Stefan Jansen from Leuven in Belgium. Stefan, welcome. There's a lot of interest. There's been a lot of interest for many years in stem cells therapies. What about the current status? What's happening?
Well, Filippo, it's an interesting field, and it's a highly challenging field that is, as we see now at the ESC meeting here, coming of age a little bit. And there are some new clinical trials that really try to move the field forward. We've been working with cells in patients with acute myocardial infarction for many years, as you know, and the trials have shown variable results. The overall theme and the idea is that we should predominantly focus on patients with reduced left ventricular function that have an impaired prognosis and try to factor in cells in the treatment of those patients. The question then is, when do you do that? Which cells do you use? And there's a flurry of new data that is coming to us at this meeting that were very interesting and revealing. I would say that the majority of data really point towards the role of cells in trying to modulate the progressive decline in LV function in those patients that are at high risk and that are associated with high mortality and morbidity.
We had the first generation of stem cells, mainly bone marrow stem cells. Now there are new generations, and in particular cardiac stem cells and mesenchymal stem cells. What is your feeling about these new cell types?
The idea behind these refinements is to try to come up with a better cell product and see whether we can better address the need for improvement in the functional decline in these hearts. The mesenchymal stem cells are often derived from the bone marrow, not necessarily, but they have been derived from bone marrow. And at this Congress, we have seen new trials where these cells have been manipulated to make them even better in trying in the repair process. There's some interesting data that are coming to light in that respect. These cells that are being isolated from the heart have a little bit of a mixed phenotype. They resemble in some way mesenchymal cells in that they secrete a lot of paracrine factors that could be helpful in trying to reduce damage, in trying to improve angiogenesis, and not necessarily solely to make new cardiac muscle, mostly paracrine cytoprotective effects. There is so far no trial that is comparing those cells head to head. So what we have today is trials that are either focusing on one cell type versus the other. But I'm sure that in the years to come, and there are some protocols that will start soon, there will be combo treatments with those two different cell types.
I think it has been funded by the NIH in the States. Well, another choice is between autologous and allogeneic. What are your gut feelings about it?
Also, this is a very pertinent question, and as the field is moving and we're learning more about the possibility of allogeneic approaches, this is maybe a great opportunity for us to try to embark with pharma and with biotech companies in larger scale trials. It is very hard to perform autologous approaches in large numbers of patients. You know how we are trying to desperately to get to the necessary numbers in order to have clinical outcome trials with autologous cell approaches. If a allogeneic approach would be equipotent, that is for sure a much better way to embark on large-scale trials. We have seen the first communications of allogeneic mesenchymal cells at this meeting today, and the results are really promising and should invite for larger scale efforts to detect what the value of these cells are.
Stefano, you're defining a clear picture. You're saying, well, mesenchymal stem cells perhaps are better, and an allogeneic approach perhaps offers some advantages. What about the patient type? Would you start with acute MI, chronic MI, or non-ischemic cardiomyopathy?
That is a great challenge for all of us. We need to understand what it is that the cells do in patients at different time points of administration and in different patient phenotypes. We don't know in the acute setting of a myocardial infarction what the best milieu is to put the cells into. If you do it too soon, it may be a very hostile milieu and the cells may not work well. On the other side, cells may not persist long enough to have a prolonged effect. If you give them too early, we just don't know. We need to come up with smart ways to identify the profile of these cells and the possibilities that they can have at different time points of administration. The type of patient I would think we would need to go for patients with the highest unmet clinical need. And for sure, these are patients with depressed left ventricular function of non-ischemic and ischemic origin. And despite great trials like Paradigm, the event rate in these patients in the active treatment groups remains high enough to really warrant better treatments, and cells may be one of them.
So the chronic phase more
Chronic
than
phase, right.
acute phase. Another interesting field is inducible stem cells. Do you think there's a future for inducible stem cells in our field?
It's a hot topic, and it's a hot topic in research. We can reprogram cells quite effectively in the lab, in vitro, in some animal in vivo models been shown in the mouse model, but so far to translate this to the in vivo situation in a patient remains a formidable challenge. The problem is to really redirect them in a safe and effective way, and that is still something that is far ahead of us, I guess.
What about other approaches like the cellularization of the organ and the rebuilding over a scaffold? Do you think is something reasonable to consider in the pipeline?
Absolutely, you're absolutely right. I think cells is one part of the equation. We need scaffolds and smart ways to try to build the best milieu for cells to reside. There are some interesting trials and new approaches where people combine tissue-engineered scaffolds to have a better retention of cells. That's clearly been shown to be effective even in early clinical trials, and the group of Philippe EHJ is doing pioneering work in this field. So in my opinion, there's definitely a place for smart tissue engineering efforts to combine with cells in order to repair organ function, absolutely.
Another interesting approach is when stem cell biology comes together with perhaps gene therapy. There are some attempts to treat Duchenne disease by getting stem cells from these patients, then genetic editing and reinfusion of these cells. Well, this not for tomorrow, perhaps it's for after tomorrow. But do you think that these two fields, cell therapy and gene therapy, can come together?
Absolutely, and Duchenne is a nice example of a prototypic disease that would lend itself towards the first translational studies in that respect, because these kids have a horrible, outcome. I think also about a lot of the congestive heart failure phenotypes that may have a genetic underpinning, and where this sort of approach would be the way to go rather than just infusing cells into these patients.
Well, we are coming to the end of this interesting conversation. Well, just one last shot. If we have to design now a trial with stem cells, what would be your protocol?
I would focus on patients with impaired LV function, who we know, despite the best of therapies that we can offer, have a progressive decline over the years with a bleak outcome, and try to modulate this course by infusing cells of different origin, but definitely patients with more advanced disease.
Well, thank you. It's been a very interesting conversation. Thank you again.